All right, I will go ahead and welcome everyone to Aric Anloague's thesis defense. I've had the privilege of serving as Aric's thesis advisor and laboratory supervisor over the last year and a half or so. Aric, I would have to say, is an exemplary thesis student. For those of you who have not had a chance to interact with him in this capacity, he has made my job unbelievably easier over the last year and a half of him being involved in this project. As I look at the list of participants, attendees of this meeting, it's many people from off of campus that Aric has interacted with, and that is actually a great sign of the collaborative nature of this project. Aric jumped into a project that had gotten started only about 6 months before he arrived in the lab. It was a brand new line of investigation for us with basically no preliminary data. And he took on the risky approach of a harebrained idea and has driven it now to the first publication coming out a few weeks ago. And at least one more publication coming out of his thesis work in the near future. He's been collaborating with Nick Moldovan at the VA Medical Center on some of this work. He's also been collaborating with Dr. William Thompson at IU School of Health and Human Sciences in the mechanobiology core. And Aric's thesis committee has been myself, Dr. Julia Hum, and Dr. Brian Larson, who unfortunately could not be with us today, but emailed about an hour and a half ago to give his well wishes to Aric. It was great to see that email from Brian. The last thing I will say is that Aric envisions a career as an educator scientist in the future. He is applying for PhD programs, and throughout all of his work in the Bone and Muscle Research Group, he has been adjuncting in the teaching capacity in the College of Arts and Sciences with in ecology class and also helping in the biostats class on the undergraduate side. He is a well-rounded individual, and I have every certainty that he will be successful in today's defense, and moving forward, he will join the ranks for an academic career. At least we haven't dissuaded him of that affection as of yet. Without further ado, Aric, I will turn it over to you. We look forward to hearing what you have to say.
All right. Thank you, Dr. Lowry, for that introduction. I really appreciate your kind words, and I appreciate everyone for joining me here today for this defense, this last sort of step for my master's program here at Marion. And so, like Dr. Lowry said, my name is Aric Anloague. I am in the BMS master's program here, and this is my thesis work looking into the mechanical stimulation of soft tissue cells in vitro, regulating pro-inflammatory cytokines. And so I've been working on this for, I guess, about a year and a half now, maybe a little bit longer. Under Dr. Lowry as my PI and in the Bone and Muscle Research Group. And so I hope you enjoy my presentation here and hope you learned something, and I appreciate you all being here. So starting off with this, I want to talk about some of the clinical correlations and connections that we're going to be discussing here. And in particular, I want to talk about sarcopenia. And so sarcopenia is characterized by decreased muscle mass and strength and is associated with increased risk for disability, loss of independence, and mortality. And so I found this figure here, and I think it's a pretty good job of kind of highlighting some of the complex natures and complex burdens of sarcopenia on public health and on individual life for those who suffer from sarcopenia themselves. And so I want to focus your attention here to this comorbidities portion here. Some of the comorbidities of sarcopenia, which can either cause sarcopenia or sarcopenia can lead to, are osteoporosis and osteopenia, obesity and diabetes, and cancer as well. And so sarcopenia can kind of be intertwined with lots of different ailments that people suffer from throughout the health spectrum here. And all of these things, if you add sarcopenia to the mix, we can increase loss of independence, increase risk of falling, and these can of course all lead to hospitalization, ambulance care, institutionalization, all these healthcare costs and decreased quality of life costs that are associated with sarcopenia. Considering these comorbidities and some of these other ailments that often occur with sarcopenia, it's difficult to look at some of the prevalence of it, but we do our best, and we have seen that generally what we see is an increase in sarcopenia in both men and women with old age in particular. And so this figure above here is from 2013 and shows the gradual increase in pre-sarcopenia and sarcopenia with old age with each decade. And so what we end up seeing is that approximately 10% of adults worldwide over the age of 60 suffer from this. And so that's a great percentage of the population that this affects. And so this is definitely something that we need to consider and look for potential treatments with this. And as discussed before, there is, of course, the increased risk of disability. We see this with increased fall risk that can lead to fracture, that can lead to other sorts of injuries as well. And of course, decreased quality of life and independence that we see with this old age. So moving forward, I want to talk about what might cause some of these characteristics that we see with sarcopenia. And so again, we're looking at decreased muscle mass, decreased muscle strength. And one of the biggest things that we look for with muscle weakness and loss of muscle mass is this term known as muscle wasting. And so muscle wasting we see is the death of muscle cells and is often a result of a chronic inflammation brought on by increased levels of the pro-inflammatory cytokines, interleukin-6 and tumor necrosis factor alpha, TNF-alpha. And we also see decreased levels of insulin growth factor 1. And so all these effects that we're seeing can cause this chronic inflammation and can eventually result in loss of muscle wasting or loss of muscle mass by muscle wasting, excuse me. Looking specifically at this study here, this is the Health and Aging and Body Composition study. We saw the research that performed this saw that in community dwelling elderly individuals, we see higher levels of TNF-alpha and IL-6 in correlation with lower quadriceps strength and mass. And so the more TNF-alpha and IL-6 that we have, lower mass and strength that we have in the quadriceps muscle. And so this is exactly what we're observing with these kind of sarcopenic tendencies here. Additionally, we see older women with high levels of IL-6 and low levels of IGF-1 show decreased walking functionability and increased mortality. And so this is, again, the same trends that we're seeing here. As we get older, we have this chronic inflammation that may be caused by increased levels of interleukin-6 and TNF-alpha, leading to some of this muscle wasting that can occur with sarcopenia itself. So looking specifically at interleukin-6 and TNF-alpha, I want to talk about some of these mechanisms that may cause this muscle wasting. And so interleukin-6 activates the JAK-STAT signaling cascade, which has a number of effects, including pro-inflammatory effects, angiogenesis, cell survival, proliferation, and transformation. And those specifically are related to cancer itself. But of course, we're more focused on this pro-inflammatory nature of it because this is what can result in some of these muscle wasting effects that we see. It's also important to note that the activated pathways of IL-6 are dependent on the duration of expression. And so what I mean by this is as different activities will cause different levels of IL-6 to change. For instance, with exercise, we see an increase in IL-6. But this has mostly metabolic and immunological effects that actually help the body. But with prolonged activation, this is where we kind of see that chronic inflammation. And we see this in elderly individuals as well as individuals with cancer. So we get this chronic activation of IL-6 that produces this chronic state of inflammation. TNF-alpha also is pro-inflammatory in nature. But it activates the ubiquitin proteasome pathway, and this can result in apoptosis and decreased protein synthesis. And so this specifically can cause some of the cellular effects that we're seeing with muscle wasting. We're seeing death of cells, death of muscle cells, and decreased protein synthesis. And so overall, what we're looking at is increased inflammation, increased apoptosis of muscle cells, and decreased protein synthesis is resulting in this muscle wasting that can occur during sarcopenia here. So what do we do to treat this? How can we combat some of this weakness that we're seeing? One of the most common things that we see are nutritional and endocrinological agents, excuse me, for secondary sarcopenia. So this is sarcopenia brought on by comorbidities such as obesity and diabetes and cancer. And these are okay for those sorts of circumstances, but we don't have a great treatment for primary age-related sarcopenia that develops without a comorbidity. And so one of the biggest things that we see is resistance training to treat this. And so by, of course, increasing resistance training, we can increase that muscle mass and increase that strength. The caveat, of course, is that with sarcopenia affecting mostly elderly individuals, it's often harder for elderly individuals to keep a resistance training regimen as they age, because it becomes harder for them to perform such activities. And so what else is there? Well, not really much. And so the most of the advice that we see is that you decrease risky activities. And so this is where we see that decrease in quality of life. We see avoiding staircases, avoiding long walks, and situations that may put them at risk of falling due to their muscle weakness. So we need to address some of these issues. We need to figure out better therapies for the elderly, for individuals experiencing this age-related sarcopenia that we're observing. So this brings us to this technique, soft tissue manipulation. So soft tissue manipulation is a broad category of manual therapy in which treatment is administered by hand or by rigid instrument. And so we see this a lot in physical therapy. We see it in osteopathic techniques as well. There are several different types of soft tissue manipulation. But what we're seeing with soft tissue manipulation in particular is we are mechanically stimulating cells. And so soft tissue manipulation is a type of therapy that uses this mechanical stimulation to affect biological processes via mechanotransduction pathways of these mechanosensitive cells. So today we're going to focus on three different types of mechanosensitive cells. These are fibroblasts, which when mechanically activated will regulate myoblast differentiation. Skeletal myocytes themselves, so when we mechanically activate them, we increase satellite cell activity via nitric oxide signaling. And so satellite cells themselves can drive formation of new muscle fibers. And so when they're mechanically stimulated as well, they will increase human growth factor. So what we see with soft tissue manipulation is we're activating these cells, we're activating these biological pathways, and overall, what we want to see is reduce inflammation and reduce levels of pro-inflammatory cytokines while building muscle mass and strength as well. So to establish this, a group created a rodent model for sarcopenia here. And in this rodent model, rats' hind limbs were suspended for 14 days. And what they observed was a decrease in muscle weight, as you would expect. Specifically, they looked at the medial gastrocnemius and the soleus. And so we see in this chart above, from baseline, the longer that they had their hind limbs suspended, the more muscle weight percent loss they had in both the gastrocnemius and the soleus. More specifically, they wanted to look at, if we're looking at the cross-sectional area, we want to look at how we can affect this change. And so we see see this loss of cross-sectional area in gastrocnemius. We're seeing a loss of muscle mass in the gastrocnemius with the hind limb suspension, this HLS. When these rats are allowed to reambulate on their own, we do see a relative increase of this muscle mass on their own as they are allowed to walk around in their cage. We are going to see recovery that will occur on its own. But when we introduce soft tissue manipulation to this and we add our treatment to it, this is what we want to see. We can see an increase in recovery of muscle mass following hind limb suspension. So we saw up to, or this group saw up to a 50% recovery in muscle mass following hind limb suspension. Additionally, what was interesting that this group discovered was that not only are we seeing an increase in recovery in the muscle mass of the treated limb, so the limb that received the soft tissue manipulation, but we're also seeing muscle mass recovery in the contralateral limb as well. And it's about the same in terms of the percentage of recovery in this muscle mass. And so what this tells us is that we're seeing some systemic effects brought on by this soft tissue manipulation. And so this kind of led us to our idea that maybe it's the cytokine levels that are driving this. Maybe by changing these cytokine levels through soft tissue manipulation, we can produce some of these systemic effects that we're observing, that this group had observed with the addition of the soft tissue manipulation treatment. So we wanted to translate this to an in vitro model. And so we used this FlexCell FX5000 tension system in collaboration with Dr. William Thompson at IU School of Medicine here. And with this system, what it does is it elongates the cells based on the resting length of each of the cells. And so I have a quick little video here that I just took on my iPhone, so it's not the best quality, but it kind of shows how these plates are able to move in this vacuum manifold. So you can imagine what's happening here is these plates are resting in this manifold, and a vacuum force from underneath is pulling down and causing these flexible membranes to be pulled down. And when you pull these membranes down, you are elongating the cells that are fixed to the membranes themselves. And so we're with this system, it comes a computer to monitor that we can actually program it to control specific lengths of elongation based on the resting length of each cell. This is also a beneficial protocol to use because as these cells are being elongated and stressed or stimulated, we can easily collect conditioned media that the cells are releasing, whatever they're releasing in response to the mechanical stimulation into. And so this conditioned media is what we will analyze in terms of looking for these changes in cytokine levels as caused by some of this mechanical stimulation. So another group has started this protocol and looked slightly into this and developed these strain profiles that we are going to use, or we have used in our model. And these profiles are cyclic short duration strain, and so this is indicative of a repetitive motion injury. And so one of the CSDS profiles that we use elongates the cells from 0%, so resting length, up to 10% beyond its resting length every 1.6 seconds, and this protocol goes for 8 hours. And so this isn't necessarily identical to a sarcopenic disuse atrophy, but what it does create is a repetitive motion atrophy in these cells. And so we're seeing some similar effects in terms of trying to create an injury like strain for this model here. The other profile that we use elongates cells starting at 10% of beyond elongation or beyond their resting length, and then goes up to 16.6% beyond the resting length. But it again repeats every 1.6 seconds for 8 hours. And so we allow these cells to undergo this atrophy for 8 hours. And then with some cells, we give them a break before we introduce them to a different stimulation. And this is our model for soft tissue manipulation, our acyclic long duration stimulation. And so this is a very short protocol and lasting only a minute. But this is originally based off the group that developed this protocol, based their protocol off of the soft tissue manipulation technique myofascial release. And so they observed videos of osteopathic physicians performing myofascial release, noted about how long they were applying pressure for and how much they were elongating the soft tissue, and developed this model based on that. And so this model increases to 6% beyond resting length and holds there for 60 seconds, and then has a gradual decline for the last 4 seconds of this procedure here. And so this is our model for soft tissue manipulation. And so our goal as a lab group was to recreate these models, prove that we can use these models effectively, and analyze our cytokine levels that are released into our conditioned media based off of these models. And so what we wanted to do is we want to measure these cytokine levels that are produced in the conditioned media as a result of this mechanical stimulation. But we needed to figure out kind of what the best way to measure these cytokine levels were. And so this was actually my capstone project that I did for the BMS program here, where I used these 3 different techniques, and I wanted to see what the pros and cons were of each of these techniques. And so briefly, I've summarized kind of the points here. One of the protocols that we used was a membrane-based cytokine array. This is a protocol in which many analytes are blotted onto a membrane, and the membrane is allowed to incubate with the conditioned media, and then so the higher concentration of the cytokine in the conditioned media, the more binding that we occur. And so we can kind of compare this to a Western blot. So you'll see on the next few slides here, you'll see a figure from these membrane arrays. But the darker of a blot that we see suggests a higher concentration. So the pros of this is that we can analyze the most cytokines at once. We have a kit that actually analyzes over 100 cytokines at once. But the cons, of course, are that it is less sensitive and specific. The next technique that we use is the cytometric bead array. And so this is an assay based on flow cytometry. And so there are these beads that are conjugated with the antibodies for specific cytokines that we're looking at. And again, we allow these beads to incubate with the conditioned media that we are recovering from our samples, and then we put them through a flow cytometer, which then will analyze the relative frequencies of each of these positive hits of beads with these cytokines. And so this is a really high sensitivity. We can actually analyze concentrations of cytokines in the conditioned media down to the picogram per milliliter level. So it's very highly sensitive, and we can analyze multiple cytokines at once, but it is kind of a costly procedure there. And then finally, there's ELISA, which is one more people are relatively familiar with, which also has high sensitivity down to the nanogram per milliliter level, but you can only analyze one cytokine at a time through these kits. So like I said, we're starting off trying to replicate some of the data that we had seen by the group in Arizona that developed the CSDS and ALDS profiles. And so we started with our fibroblasts. And so this is an example of a fibroblast membrane array that we used to detect some of these cytokine levels. And so each pair of dots represents specific cytokines, except these top two sets and this bottom one here represent our reference spots here. But as we look at some of these in particular, I'll draw your attention to IL-8 here. We see a relatively similar density with IL-8 in the control and the injury strain, but then we see a reduction in IL-8 when we add our ALDS stimulation, which again is our soft tissue manipulation like strain here. So this is less sensitive. We did see some of these trends, but we wanted to make sure that we're being specific, and so we looked towards the cytometric bead arrays and the ELISAs, which did show us more positive and specific results. And so looking specifically at what we were analyzing, looking at specifically IL-6 and IL-8, what we saw was an overall decrease in expression of these pro-inflammatory cytokines with the addition of the ALDS profile, regardless of which CSDS profile that we used. And so as you recall, we had two different CSDS profiles, but both of them showed a decrease overall in the expression of these pro-inflammatory cytokines, a significant decrease with the addition of this soft tissue manipulation-like technique. We wanted to be sure that what we were measuring is what we were measuring, and that the fibroblasts were producing these cytokine levels that we were wanting to see. The bead array assay actually also measured for interleukin-1 beta, interleukin-10, interleukin-12, P70, and TNF-alpha, but we only were able to detect IL-6 and IL-8. And so we performed some RT-PCR to show that in unstrained conditions, fibroblasts are only expressing these genes for these interleukin-6 and interleukin-8 here. And so we confirmed that this is what fibroblasts are capable of secreting in terms of cytokine release. Continuing on, let's discuss kind of what we saw and what these roles of these cytokines may be. And so we did see a reduction in IL-6, and we want to kind of understand what that is telling us in terms of how this may affect muscle and how this may affect sarcopenia. And so the role of IL-6 appears to be biphasic. And so, like I said, it's dependent on the duration of exposure. And so early on, it can be important for myoblast migration, which may suggest that it would have a positive effect on muscle mass. But as IL-6 remains in the system, it'll lead to that muscle wasting and atrophy that we described earlier via the JAK-STAT pathway. In a brief experiment, muscle recovery for IL-6 knockout mice subjected to hind limb suspension took twice as long as the wild type did. And so without IL-6, you will see that muscle recovery does take long. And so this kind of highlights that need for IL-6, but we just don't want too much of it. When you reduce IL-6 after soft tissue manipulation, we may see some of this reduction in this muscle wasting that can occur from the chronic inflammation caused by IL-6. These results of IL-6 reduction were previously reported by Meltzer and Stanley. This is the group that produced these CSDS and ALDS profiles, and so we have replicated their results with our experiments. Looking at IL-8 in particular, it is important for neutrophil recruitment. It's pro-inflammatory in nature, and it will remain active as long as it is expressed. And so if we can reduce IL-8 after soft tissue manipulation, we may be able to reduce inflammation as well. And the results that we have discussed with this IL-8 reduction is new and novel. We are the first to report this observation. We wanted to expand our work a little bit further, and so we extended the cell types that we were looking at and subjected to this mechanical stimulation. So following the same protocols as the fibroblasts, where we had the CSDS profiles followed by ALDS profiles to stimulate that soft tissue manipulation, we subjected C2C12 mouse myoblast cells to this procedure here. And briefly, on the right, you'll see this is our membrane array. We tested for a lot of different cytokines. There are so many to report on. I wanted to highlight a few that we saw we saw significant changes only with the n equals 2 here. So it's a small sample size, but we did see significant change in these three specific cytokines here. And so what we saw was that with the addition of this ALDS, so during the CSDS and ALDS strain, we saw that angiopoietin-1, MCP1, and IGF-BP5 were all decreased with the addition of soft tissue manipulation. So briefly, I listed here a couple of the roles of these cytokines, but we will need to confirm these results with a ELISA or beta ray, and we want to look a little bit more closer and increase the power of our study by repeating some of these protocols. We also looked at human skeletal myocytes. In a similar manner, we followed the exact same protocols with this, and briefly we saw that soft tissue manipulation has reduced DKK1, ENA78, and FGF7 with the addition of this soft tissue manipulation like treatment. Again, we only have an n equals 2 here, and so we need to repeat these protocols to kind of increase the power of our study. We're a little bit limited this spring and summer, obviously with the coronavirus pandemic here. So we were unable to perform as much as we wanted to, but we have enough to kind of get us started here, and we'll of course continue with our collaboration as we move forward. So finally, I want to kind of talk about where we want to go with this study, what our next steps are, and how we're going to continue. I think the obvious one here is that we'll repeat these C2, C12, and myocyte procedures to increase the power of these studies. We'll also look at them via ELISAs or via beta rays to see if we can get some more specific and sensitive quantifications of those changes in cytokine levels that we're seeing. But I want to move forward beyond that. I want to talk a little bit about other potential insights, because this project is ripe with opportunity here. And so one of the areas that I'm excited to expand to are expanding this model, to adipocytes. Adipocytes, of course, are fat cells and are present in the soft tissue, and have would be affected by soft tissue manipulation mechanically. And they're already known to secrete these cytokines, TNF alpha and IL-6. One of the biggest things that we see is that TNF alpha and IL-6 secreted by adipocytes can cause chronic inflammation, especially in obese and insulin-resistant individuals. We actually see that IL-6 will induce insulin resistance in a specific type of adipocyte. This is performed by other researchers as well, but of course is related to our work here. And then we saw that IL-6, IL-8, and TNF alpha are overexpressed in human fat cells from insulin-resistant individuals. And so this is bridging our gap between mechanical stimulation and sarcopenia towards mechanical stimulation, and maybe even something some metabolic disorders like obesity and diabetes as well. It's also been shown that reduction of TNF alpha can improve insulin sensitivity. So maybe this can help with diabetic individuals if we can reduce TNF alpha levels with soft tissue manipulation. But of course, we'll have to analyze those adipocytes to of course confirm that. So adipocytes, of course, brings us to a totally different avenue than muscle-specific roles, but there's lots of different areas that we can look into in terms of looking at adipocytes being affected by soft tissue manipulation and mechanical stimulation. Lastly here, I forgot I had this point here, but mechanical stimulation of adipocytes has been shown to decrease adipogenesis. And so, again, this is similar to some of the trends that I've already discussed here, but mechanical stimulation is beneficial and can have lots of different effects in adipocytes. Moving forward to other areas that we can extend this model, I put this "bones are beautiful" line here for Dr. Lowry and Dr. Hum, who are, of course, the bone experts here, and then love their bone research here. So this is some preliminary work that we actually have done in our lab. Taylor Highland and Ting have helped me with this in particular as we move forward. What we're doing is we're exposing this line of osteoblast precursor cells, so these W2017 cells, which are osteoblast precursors, and of course osteoblasts are bone-forming cells. We are exposing these cells to the conditioned media collected from our fibroblast flex runs, and so we saved that conditioned media that we analyzed for cytokines, and we are exposing these cells to this conditioned media to see kind of the effects of this differentiation. Can we get these cells to become osteoblasts with exposure to our conditioned media? And so far, what we have seen is very positive. Exposure to these cells has increased levels of osteocalcin, which is an osteoblastic marker. We analyze this by QRT-PCR. And so what we see is that with no conditioned media, we have this baseline here. Is very similar to the control conditioned media. So the conditioned media of cells that were not did not undergo the flex protocols, either the CSDS or the CSDS plus ALDS. When we added that CSDS plus ALDS soft tissue manipulation like conditioned media to these precursors, we saw almost a twofold increase in osteocalcin, suggesting that we are getting some differentiation into osteoblasts, which may suggest that this soft tissue manipulation can be a potential therapy for things like osteopenia and osteoporosis. This is further expanded to an ex vivo model here, where we exposed neonatal tibia explants from mice to this same conditioned media that we used in the W20 protocols from the fibroblast flex runs. And in this, we were looking at the expression of the bone resorption markers and bone formation markers, P1NP and CTX here. And so P1NP is representative of our bone formation marker, and CTX is representative of our bone resorption marker. And so when we expose these tibia explants to our conditioned media that has undergone this ALDS strain, this STM strain, we are seeing an increase in bone formation markers and a decrease in bone resorption markers. And so again, this maybe suggests that bone anabolism can be activated by soft tissue manipulation here. And so this connects, of course, to lots of the other bone research going on in our lab group, and hopefully we can continue pushing this forward. I believe Taylor's going to do a lot with this work and continuing this on when he does his thesis as well. Another area that we are looking into, this is in collaboration with Dr. Moldovan over at the VA and the 3D Tissue Bioprinting Core Lab, where we're creating these three-dimensional models of these cells. And so far, all of our work has been in a two-dimensional monolayer of cells. And so when we think about the cells on these plates, it's only in one layer of cells, which is not necessarily indicative of a tissue system, and mechanical strain on a single layer of cells is going to be much different than mechanical strain on a three-dimensional model. And so using this 3D tissue bioprinting core, we are printing these cells into a gelatin matrix, giving them three-dimensional structure. Down the road, we may even do in co-culture, where we have fibroblasts, C2, C12 cells, and myocytes all in the same membrane. in this three-dimensional tissue model. And then what's kind of exciting is we're using this new compression system that we are trying out right now up in our Evans Center laboratory here, which is similar to the FlexCell system. It's through the same company, the FlexCell company, but it is the compression system. And so in the previous model, what we had been using was the tension system, which elongates the cells by pulling on them from below and stretching them out. This tension system presses the cells together, which can cause elongation, but it provides compressive forces instead of tension forces on these cells. And so this may be a good model to use too, because lots of soft tissue manipulation techniques involve these compressive-like forces as well. So we're still in the process of kind of establishing what these models will look like, how we can effectively test these models. But this is a very exciting next step that we're in the process of taking here. And so I have a little video, which is less exciting than the other video that we had, because you can't really see what's going on in the wells, but you can definitely hear what's going on. It's so what's happening essentially is that there's a movable piece at the bottom of each of the wells that is pressing up into the cells against almost like a pedestal, and then compressing the cells that way. And so this is where the next steps of our project are going in terms of the in vitro models. But we also want to expand this to more in vivo models. And so our goals are to recreate the rat hind limb suspension model. And so we'll follow the similar protocols that the previous group has done. We'll do 14 days of hind limb suspension, and there will be a treatment group undergoing soft tissue manipulation. And of course, the control will not have the soft tissue manipulation, will reambulate on their own. But our goals with this animal model is to analyze specifically some of the cytokines that we observe change from all of our cells. So IL-6, IL-8, and hopefully TNF-alpha if we get those adipocytes analyzed. We'll look for these changes in levels in the serum of each of these groups, and ideally, the animal model will reflect our in vitro results, showing that soft tissue manipulation overall will decrease levels of pro-inflammatory cytokines. So with that, I want to thank everyone who's been involved in my process. It's a lot of people, but I know, particularly Lottie, Ting, and Taylor from the BMRG. They have all helped me specifically with my experiments. They have all been a huge part in helping me make sure that I get all my work done in on time, and of course our great friends. The BMRG in general, the whole bone and mineral research group, bone and muscle research group, has been very supportive of me and has been easy lab mates to work with, which is of course what you want in a lab setting. You want people that you can get along with and that you have little conflict with. I want to thank Dr. Julia Hum and Dr. Brian Larson for serving on my thesis committee and taking the time to review my work. Even though Dr. Larson couldn't be here for this defense itself, he's still taking the time to look over my written thesis as well. I want to thank Dr. Thompson and Dr. Moldovan for allowing me to invade their spaces and helping me perform some of these techniques that we are unable to perform here on Marion's campus, and for their collaboration. And then lastly, I want to thank Dr. Jonathan Lowry, who has been an excellent mentor to me, helping me find myself, find my career goals, and being excellent support along the way. To the right here, I have this photo from ASBMR 2019. This is the American Society for Bone and Mineral Research. I didn't perform at this conference, but it was an excellent experience to learn about some of the bone world, and it was an excellent experience traveling with some of our friends in the BMRG here. So with that, I guess I will open it up to questions. I have citations here if anyone wants to look at some of this. Yes, Jonathan, that was definitely a arcade gallery. It was like almost like a Dave and Buster's down in Florida. There, as in Orlando. That's where the conference was. But it was a fun time.
Aric, thank you for the presentation. Is my audio coming through?
Yep.
Okay, great. I noticed there were a couple of other collaborators that jumped on the call while we were going. I want to specifically call out Dr. Terry Lagmani from IU School of Health and Human Sciences, who has been involved in this project for a long time now as we think about its development. And then Dr. Jesus Delgado Calle, who is now at University of Arkansas for Medical Sciences in Little Rock. He was the collaborator for our ex vivo tibia explants. Very appreciative for everyone for pushing this project. We will have a private defense in just a moment. I see, Aric, that there's a question for from Dr. Gelfman in the chat. And if anyone else would like to ask a question, you can either put it in the chat bar or just type in Q in the chat and we will call on you.
All right, Dr. Gelfman, thank you for that question here. So he was asking how long was the soft tissue manipulation performed and how long was it simulated in the lab? And so in our in vitro experiments, the ALDS strain is how we performed our soft tissue manipulation. What we did was we exposed ourselves to the injury profile, the CSDS profile for 8 hours. Then we waited 3 hours, let the cells rest for 3 hours. And then we exposed them to the ALDS soft tissue manipulation, which was actually only 1 minute long. So it's kind of incredible that we're seeing some of the results that we have seen with such a small profile for treatment, but we're seeing some very significant results across the board here. So excellent question. I appreciate the question. When we analyze our conditioned media, I don't know if I said this, but it's a few days after, so we allow the cells to kind of relax after the stimulation in the flex cell machine. So for our fibroblasts, we waited 96 hours to collect a conditioned media, and for our C2C12s and our myocytes, we waited 72 hours before collecting the conditioned media. I see another question here from Dr. Delgado-Calle, and he asks, any idea how our conditioned media decreases CTX in the ex vivo organ cultures? That is absolutely one of the things that we want to investigate further. We are understanding that we're seeing these differences, but we aren't necessarily understanding exactly how this is occurring. So that's definitely something we're going to explore as we move forward with our differentiation assays with the osteoblastic precursors and our ex vivo explants as well. All right, so next question here. So the question says, it's my understanding that the amount of adipogenesis in adults while present is low. So what amount of impact are we trying to see when we expose adipocytes to these pathways? That I'm also not certain about. The adipogenesis definitely is something I'm less familiar and have not studied as much. But as we dive into the literature and explore the adipocytes in terms of their response to mechanical stimulation, we want to see what some of these effects are in terms of adipogenesis itself or reduction of some of the inflammatory natures of adipocytes. And to me, I'm more interested in if we can reduce levels of TNF alpha and IL-6 expression from adipocytes than adipogenesis in particular, but it was definitely an interesting area to look into as we go through this protocol here. Next question I see from Dr. Moldovan is how the implications of our work can be implemented in practice. And he gives the example for example in low gravity in space. And I think that is an excellent point that we can look into. Sarcopenia is the example that we used in this particular experiment. We see that we want to try to increase muscle mass with just something as simple as soft tissue manipulation. But in the instances of low gravity in space, without the weight of gravity, we often see increase in osteoporosis and osteopenia, and as well as some things like sarcopenia. And so in a setting like a space station or a spaceship, I bet it's a lot harder to do resistance training, especially with low gravity. And so implementing soft tissue manipulation techniques to create that sort of anabolism in muscle and in bone may definitely be beneficial as we move forward with some of these techniques here. Dr. Dellery is talking about I appreciate the kind words about the presentation. She was talking about making correlations to wasting seen in patients living with AIDS. That's something I haven't thought about at all, but I would imagine that because the mechanism is relatively outside some of the processes that we see and defects that we see with AIDS, that things like soft tissue manipulation may help in terms of reducing some of that muscle wasting that we see in some of those patients. So definitely something to explore. Yes, Dr. Lowry, with muscle loss with COVID as well. The COVID conversation has definitely changed lots of the therapeutic and clinical connections that we see. So I appreciate that. Yes. Any more questions from anyone else? Again, I appreciate all of you for attending this seminar, taking your time to watch my presentation. I appreciate you all being here, and I appreciate to all of those of you who have helped me along the way. Thanks again for all you've done.